Today, I shall be discussing about neoplasia in my four-part series. It is important to know the concept of neoplasia in order to truly understand the nature of cancer. Hence, it is important for us to know, regardless if you want to become a surgeon or a physician. Now, to begin with, we should start with the definition of neoplasia. So, what exactly is neoplasia? Neoplasia is defined as an abnormal mass of tissue, the growth of which far supersedes that of normal tissue, and is uncoordinated with the said normal tissues and persists in the same excessive manner after cessation of stimuli which evoke the change. Now we shall be moving on to the general features of neoplasia. What exactly defines it? What truly characterizes it? So to start with, let's move on to the origin. Now, all neoplastic cells arise from cells with normal proliferative capacity. This is a general rule. Now, they are irreversible as they persist even after the stimulus is reversed. The same stimulus which is causing this change in the first place. Once you take it away, it's still going to continue to multiply in its same excessive, uncontrollable manner. Now, often it can be caused by genetic disorders due to mutations, which are permanent genetic changes. So now genes which can regulate cell growth or control apoptosis or DNA repair are often mutated or highly damaged. As a result, there is no checks in place for cell growth, apoptosis, or DNA repair. So this can cause cells to wildly multiply without any control in place. Now, sometimes a carcinogenic stimulus can be involved, which is responsible for uncontrollable cell proliferation. Now, all these genetic alterations which happen, they're heritable. So suppose a mother cell happens to have an alteration to its genetic code. This can be passed down to the daughter tumor cell. The exact same copy of the mutation will be passed down. As a result, all daughter tumor cells will have these genetic mutations. Now, neoplastic cells are monoclonal. This means that all cells within an individual tumor arise originally from a single cell which had that genetic change. So suppose one cell in normal tissue had a genetic change. Now, there's no checks and balances in place for the cell to multiply. So it's going to continue to multiply out of control. Eventually, you're going to have that individual tumor where all cells from that tumor possess that same genetic change, which they inherited from the mother cell. Now, neoplastic cells are also autonomous. So since they're excessive and have unregulated proliferation, they do not obey regular controls. They work independent of physiological stimulus, and they still depend on the host for nutrition. Now, you might ask, if they still depend on the host for nutrition, how are they autonomous? Well, apart from nutrition, which it derives from the host like a parasite, everything else, all its normal functions, they act independent of the host. So if the host tells them to stop multiplying, it will not continue to stop multiplying. It will, in fact, continue its progress. And there's differentiation. Probably one of the most important features. Differentiation is defined as the extent to which tumor cells resemble the original cell. Now, moving on to differentiation, but let me further explain what it is. Its extent to which neoplastic parenchymal cells resemble the corresponding normal parenchymal cells. So now there's three things you have to remember. Something called as well-differentiated cells, moderately differentiated cells, and poorly differentiated cells, right? So now, well-differentiated cells are often more mature in nature, and poorly differentiated cells are often immature in nature, all right? So since well-differentiated cells can closely resemble the original cell, this means that the cell in question since it closely resembles its parent cell, it takes its own sweet time to mature, to resemble a parent cell. However, the poorly differentiated cell does not care about how closely it can resemble the original parent cell of the normal tissue. It just cares about multiplying as fast as possible. So as a result, the development of the cell is immature, yet it is more aggressive in its nature of multiplying. So now, well-differentiated cells will always be found in benign tumors. Whereas malignant tumors, you have the full spectrum of poorly differentiated cells, moderately differentiated cells, and well-differentiated cells. All right? So let me repeat this one more time. Benign tumors will always have well-differentiated cells, whereas malignant tumors will have poorly differentiated cells, moderately differentiated cells, and well-differentiated cells. All right? Since I brought up the topic of benign and malignancies, let me classify them for you. So tumors in general can be classified as benign and malignant. Now, benign tumors are well-differentiated cells, all right? They will always be well-differentiated, and they grow very slowly. They can take up to years to reach a certain size, and they're often well-circumscribed or capsulated, and they're localized to the site of origin. The fact that it's well-circumscribed or capsulated makes it easier to localize, and it makes it easier to remove it from the body. Malignant cells can be poorly differentiated, moderately differentiated, or well differentiated, and they always grow faster. Especially the more poorly differentiated the cell is, the faster it grows. They're poorly circumscribed as they adhere to neighboring tissue and they invade them and they just latch on and they metastasize to distant tissues. All right, this is the most characteristic feature of malignant cells. They will generally metastasize to distant tissues to spread. All right. Now, to truly understand what makes a cell malignant, we have to discuss a topic known as anaplasia. So now, anaplasia is actually the failure of differentiation of cells. All right. So now, the greater the anaplasticity of a cell, the more aggressive the corresponding tumor will be. All right. So let me repeat this one more time so that you get this concept more clearly. Anaplasia is the failure of differentiation of cells. So the more anaplastic a cancer cell is, the more aggressive the tumor will be. All right. So now let's look at the microscopic level. What does normal tissue look like versus what do anaplastic cells in tissue look like? All right. So over here you have this diagram. And you have normal epithelium over here. As you can see, the cells maintain a normal cell size and nuclear size. The nuclear to cytoplasmic ratio is all normal. It's either in the ratio of 1:4 or 1:6. And cell polarity is well organized. All cells adhere to one another properly. And they're all arranged in layers. Right? Now, moving on to anaplastic cells over here. There's a complete variation in shape and size. The cells and nuclei show something known as pleomorphism, what is a complete variation in the shape and structural organization of these cells. And there's an increase in the nuclear to cytoplasmic ratio. In normal cells, it's 1:4 or it can be 1:6. In anaplastic cells, it's completely thrown off and can potentially reach 1:1. As you can see, that one cell there, the cytoplasm is almost nowhere to be seen. The nucleus occupies almost the entire cell. All right, and the nucleus of the cells are often hyperchromatic in nature, and they show coarse chromatin. Right, and we shall get to why that is soon. And there's a complete loss of polarity of cells. Now, polarity refers to how cells are oriented to one another. This affects the way they're arranged and the way they're organized. Now, if there's a complete loss of polarity of cells, this means that these cells will not sit next to one another so easily. They'll be completely disoriented from each other, and it's easier for it to break away. So this makes invasion easier. So it'll closely approach the nearest blood vessel available and invade and spread to distant sites. All right. Now, what's causing this hyperchromatic nuclei? It's something called as mitotic bodies. All right. So now, in a normal cell, it's bipolar. You find two poles on opposite sides of mitotic figures. Now, sometimes when you lose that property of forming bipolar cells, you have something called tripolar and quadripolar cells. As you can see, you find 3 poles, 1, 2, 3, and 1, 2, 3, 4. These are known as atypical mitotic figures. Now, anything more than 4 becomes so bunched up together, it's called a bizarre cell. All right. So this is where you find 5 or more mitotic spindles forming in different directions, or it's all clumped together, so it's very difficult to make out. Sometimes they just break apart from each other and drift away, and they're known as tumor giant cells. All right. So I've written them down over here. Pleomorphism. That's variation of size and shape of the cells and nuclei. It's a characteristic feature of malignancy. And it varies from large cells all the way down to small primitive cells. Now, these small primitive cells refers to how the cells can't really be differentiated from one another. And the nucleus almost occupies the entire shape and size of the cell. And it's very, very, very aggressive in nature. So it's going to constantly multiply very fast and has no time for differentiation. And there's abnormal nuclear morphology. So the excessive amount of chromatin, basically from the excessive number of poles and increased number of DNA per cell, results in the hyperchromatic nuclei. As a result, it will stain very darkly. And the nuclear size and shape is variable. Sometimes you can find large prominent nuclei, that is like in the bizarre cell and the tumor giant cells. And you find mitosis. It's like mitosis, except without an I, spelled with an E towards the end. Right? So mitosis refers to these atypical mitotic figure cells. So anything more than 2 is actually a cause of concern. Anything more than 2 mitotic spindles. It's called a bizarre cell. And as I mentioned earlier, the nuclear to cytoplasmic ratio in normal cells is 1:4 or it's 1:6. And abnormal cells can go up to 1:1. And there's loss of polarity. The orientation to one cell to another is lost, so there's disturbed orientation. And you find bizarre cells, as I discussed earlier. Large polymorphic nuclei. It's 2 or more large hyperchromatic nuclei. That's called tumor giant cell. All right. Now, we've discussed the structural changes of these cells under anaplasia. What would happen on the functional changes? What changes functionally? Now, the well-differentiated tumors generally retain the functional characteristics because they're mature enough to carry out these functions. Now, the overall function might decrease a little bit, but won't be as bad as compared to poorly differentiated tumors. Now, these well-differentiated tumors can sometimes secrete normal substances. For example, hormones, like a well-differentiated carcinoma of endocrine glands can secrete the corresponding hormone from which the original cells secreted. Sometimes they secrete normal products which you find in the body. For example, squamous cell carcinoma, which are written as SCC, secretes keratin. And sometimes this keratin, it curls up and deposits, forming an epithelial pearl, which you can often find in a cut section of a squamous cell carcinoma, which is what leads to the diagnosis of carcinoma, squamous cell carcinoma. Sometimes you find abnormal products, such as fetal proteins. Now, they are produced in the body normally. However, it's not produced comparable to, let me put it this way. It's generally not in detectable states in normal adult cells. So when there's a sudden increase in these products, that's when it's a cause for concern. Hence, it's used as a marker for any abnormal neoplastic cells. Sometimes some cells secrete ectopic hormones. So tumors may produce hormones which are not indigenous to the corresponding tissue of origin. For example, bronchogenic carcinomas can produce ACTH or parathyroid-like hormone. So sometimes it can really, really throw off your diagnosis when you find hormones being secreted from entirely different sites. And we shall discuss this further in paraneoplastic syndromes, which will come way towards the end of this series. Now, moving on to the rate of growth. Now, the rate of growth is directly related to the degree of differentiation. So now, benign tumors, which are very well differentiated, generally grow slowly. And malignant tumors, which are not always well differentiated, they can largely be poorly differentiated, grow rapidly. And the rate of growth is also dependent on hormone stimulation, for example, or adequacy of the blood supply. Adequacy of blood supply is generally given because these tumors are like parasites. And hormone stimulation, we find some tumors such as uterine leiomyomas, which are dependent on certain hormones. Now, if it increases in size in cases such as pregnancy and decreases during postmenopausal period, it's dependent on one particular hormone. Can you guess which hormone that is? The answer is estrogen. There's an increase in estrogen levels in pregnancy, which causes an increase in size of uterine leiomyomas. And in postmenopausal period, there's a decrease in estrogen levels, hence causing a decrease in size of the uterine leiomyoma. All right. Now moving on to the balance between cell production and cell loss. Now, there's something called doubling time of tumor cells, right? So this means that the total time required for a total cell cycle to take place is actually decreased by mitosis. So by what I've written here, cell to double by mitosis, this means that for the amount of time for a cell to double in number, it's actually cut by half. And there's growth fraction. So that's proportion of cells in the proliferative or replicative pool within the tumor. Now, suppose we had a tumor over here. Let me just draw a tumor. I've got two tumors, to give an example. Suppose only 10% of the tumor is involved with replication. The rest of it just stays still. We can call this a slow-growing tumor compared to, let me label this as A, let me label this as B. Now, A is slow-growing compared to B, because in B, you have close to 40% of the tumor producing cells, like constantly rapidly multiplying. The rest stays still. So now the fact that more cells in tumor B are involved in the replicative process more than tumor A, you find that it has a much higher growth fraction. Hence, it multiplies much faster. So cell production is definitely greater than the rate of cell loss in this case. And there's the rate of tumor cell death. So when the rate of cell production and loss by apoptosis increases, there's a high cell turnover. All right? So now, if more cells continue to die out, more than cells are being produced, it's called high cell turnover because there's larger cell die out. Whereas in neoplasia, this is not the case. Cell production far supersedes cell loss. So, and there's also decreased apoptosis because sometimes the genes for apoptosis is just mutated. So now, as a result, cell production takes over cell loss. So there'll be a lot more tumor cells being produced compared to tumor cells dying out. All right. So now, what is it about malignant cells which makes it invade? Or can benign tumors invade? We shall see now. Now, benign tumors are localized, expansile masses. They do not invade, and there is no metastasis. They do not infiltrate into adjacent tissue, and they have a capsule. This capsule is made from compressed connective tissue from the extracellular matrix. This makes the tumor more palpable, makes it movable, and surgically enucleated. Right? For example, it's a diagram over here. You can see it's localized. It does not invade into neighboring structures. You can easily palpate it. You can move it around through surrounding tissues, and you can easily take it out surgically. All right. Now, malignant tumors. They show metastasis. That is a given. They do not have a capsule, so it makes it very poorly demarcated. You cannot tell the exact boundaries of the tumor involved. And they just love to invade neighboring structures. They could potentially destroy the adjacent tissue or organ. Suppose there's an important organ such as the stomach. The stomach has its normal stomach cells. Suddenly gets invaded through metastasis, it gets invaded by cancer cells. Now the cancer cells constantly multiplies in the stomach, and it slowly takes over the overall integrity of the stomach by replacing normal stomach cells, which are actually productive, with faulty cancer cells. as a result, you find that the overall function decreases. But apart from that, because of the constant invasions, you'll find that there's eventual destruction of the normal tissue, because there's nothing but invasion of cancer cells constantly eating up normal tissues. All right. Now, some tissues, however, are resistant to invasion by malignant cells. For example, mature cartilage and elastic tissues of arteries. All right. So now, as I'll discuss later, tumor cells do not prefer invading around arteries. Sometimes you'll find the closest faulty leaky capillaries, or through process of angiogenesis, they can cause new capillaries to form around them to provide oxygen supply. Yet, because they're newly formed, it's still easily permissible, so they can easily pass through and invade. All right. So mature cartilage and elastic tissue of arteries resist invasion by malignant cells. Now, malignant cells can also invade within the epithelium. This is called pagetoid infiltration. It's seen in pagetoid condition of the breast, where it invades the epithelium of the nipple. It slowly gets in through the ducts. And for an invasion to take place in a more distant site, it's called as metastasis. Now, the two most preferred routes for malignant cells is lymphatics and blood vessels. Lymphatics and blood vessels. Now, sometimes it can also invade through the perineural bundle. For example, in carcinoma of prostate and pancreas, you'll find that malignant cells invade around the peripheries of the nerve bundles till they reach another site. Now, what are the consequences of this potential malignancy? What can it cause as a result of its invasion? What are the consequences? For starters, there's functional insufficiency, because as I explained earlier, normal tissue is being replaced by cancer cells. Sometimes it can compromise vital regions. For example, brain tumors. Suppose a brain tumor compresses against the brain stem. What would happen? It can compress against respiratory centers of the brain, hence leading to breathlessness and hypoxia, eventual death. Sometimes tumors can be located in life-threatening locations, such as in the colon. This complete obstruction can actually cause complications for the colon, hence make them life-threatening. This is the end of part 1. I shall continue soon.